Immunology Insights, Basics and Beyond. Chapter 14, Immune Cell Markers and Receptors. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we discussed the roles of cytokines and chemokines in coordinating the immune response. Today, we will dive into the fascinating world of immune cell markers and receptors, those critical molecular components that allow immune cells to recognize pathogens, communicate, and carry out their functions. This chapter will cover the main classes of these molecules, their biological roles, and their importance in research. We will also explore how these markers are used to classify immune cells and highlight examples of diseases and therapies involving these molecules. So let's talk science. To introduce immune markers and receptors, the immune system depends on a complex network of surface molecules and receptors to perform tasks such as identifying pathogens, coordinating with other cells, and initiating immune responses. These molecules are vital for both innate and adaptive immunity. Key categories include cluster of differentiation markers or CD markers. They are used to classify immune cells and their functional states. Toll-like receptors or TLRs. There are evolutionary hard-coded recognition receptors essential for detecting microbial threats and triggering rapid innate immune responses. Then we have the major histocompatibility complexes or MHC molecules. They are critical for antigen presentation. And then, of course, we have the Fc receptors, the B and T cell receptors, as well as other pattern recognition receptors. They are all essential for antibody recognition and lymphocyte activation. Now let's look at these categories a little bit more in detail. Let's start first with the clusters of differentiation or the CD markers. CD markers are surface proteins that serve as unique identifiers for different types of immune cells. The combination of specific CD markers on the cell surface is used to classify immune cell subsets, a method essential in research and clinical diagnostics, particularly in techniques like flow cytometry, Western blot, and immunohistochemistry. Let's look at some key examples of CD molecules. We have CD4, which is found on T helper cells. CD4 positive T cells orchestrate immune responses by releasing cytokines that recruit and activate other immune cells. As an example for its importance, in clinical practice, CD4 counts are routinely tracked as a marker of disease severity and progression in HIV patients. Then we have CD8, which is present on cytotoxic T cells. CD8 positive T cells destroy virus-infected and cancerous cells by recognizing antigens presented on MHC class I molecules. To illustrate its role, during a viral infection, CD8 positive T cells infiltrate infected tissue to eliminate compromised cells. Next, we have CD19 and CD20 on B cells. CD19 enhances B cell activation and signaling, playing a key role in initiating antibody production and immune memory. CD20 is involved in B cell development and calcium signaling. Both are diagnostic markers and targeted in cancer and autoimmune therapies. Then we have CD14, which is a marker expressed on monocytes and macrophages. CD14 detects bacterial components such as lipopolysaccharides or LPS, initiating inflammatory responses. Then we have CD56, which is found on natural killer cells or NK cells. This marker is associated with their cytotoxic function. NK cells play a pivotal role in early defenses against viral infections and tumors. As an example, CD56 positive NK cells are often studied in cancer research for their ability to kill tumor cells. Moreover, by analyzing the combinations of markers such as CD14, CD16, and CD68, researchers can differentiate between immune cell types like monocytes, macrophages, and granulocytes, gaining insights into their unique roles in immune responses. Second, we have the toll-like receptors or TLRs. TLRs are a type of pattern recognition receptors or PRRs that detect conserved microbial signatures known as pathogen-associated molecular patterns or PAMPs. These receptors provide a first line of defense by initiating inflammatory signaling cascades in response to infection. Also here, let's have a look at key TLRs and their role. For example, TLR4 recognizes lipopolysaccharides or LPS from gram-negative bacteria, triggering pro-inflammatory cytokine production to combat bacterial infections. A good example for its potency is a septic shock, where excessive TLR activation can lead to harmful systemic inflammation. Then we have TLR3, which detects double-stranded RNA, a hallmark of viral infections, and promotes the production of type 1 interferons crucial for antiviral defense. Last but not least, we have TLR9, which binds to unmethylated CpG DNA found in bacterial and viral genomes, helping the immune system distinguish between self and non-self DNA. Third, let's look at the major histocompatibility complex molecules or MHC molecules. As discussed in a previous chapter, MHC molecules are critical for antigen presentation, enabling T cells to recognize and respond to pathogens. Also here, we can briefly look at the different types of MHC molecules. We have MHC class 1, which is found on all nucleated cells. These molecules present endogenous antigens, for example viral peptides, to CD8 positive T cells. And then we have MHC class 2 molecules, which are expressed on antigen-presenting cells or APCs, like dendritic cells, macrophages and B cells, presenting exogenous antigens to CD4 positive T cells. Fourth, we have the Fc receptors or FCRs. Fc receptors are proteins on immune cells that bind to the Fc region of antibodies, mediating immune processes like phagocytosis, where macrophages and neutrophils engulf antibody-coated pathogens, or antibody-dependent cellular cytotoxicity, in brief ADCC, where NK cells kill antibody-coated target cells using cytotoxic granules. Fifth, we have the B cell and T cell receptors, or BCRs and TCRs respectively. BCRs are membrane-bound immunoglobulins on B cells that recognize specific antigens and initiate B cell activation. TCRs are found on T cells and bind to antigen MHC complexes and are essential for T cell activation and differentiation. BCRs and TCRs are studied in flow cytometry and ELISA assays to understand clonal expansion and specificity during immune responses. Last but not least, and on the sixth position, we have the knot-like receptors or NLRs. NLRs are intercellular pattern recognition receptors or PRRs that detect microbial components inside the cell. They activate the inflammasome, a multiprotein complex that triggers the release of inflammatory cytokines such as interleukin-1 beta or IL-1 beta. With all their important functions, immune markers and receptors have pivotal roles in disease and therapy. Immune markers are integral to the pathogenesis of various diseases and have become indispensable tools for diagnosis and treatment. For example, in HIV infections or AIDS, CD4 positive T cells are targeted and depleted by HIV, leading to immunodeficiency. Monitoring CD4 levels is essential for managing disease progression. Or in B-cell malignancies and autoimmune diseases, CD19 and CD20 are central to both malignant and autoimmune conditions. Rituximab, for example, a CD20 targeting antibody, depletes B cells to treat non-Hodgkin lymphoma and autoimmune diseases like multiple sclerosis. But also CAR T-cell therapies targeting CD19 or CD20 have shown remarkable efficacy in hematological cancers. And of course, in the field of immune evasion in cancer. Tumors exploit immune checkpoint molecules such as PD-1, also called CD279, and CTLA-4, also called CD152, to suppress T-cell activity. Therapies like anti-PD-1 and anti-CTLA-4 antibodies reactivate immune responses, transforming cancer treatment. And of course in sepsis. Overactivation of TLRs, particularly TLR4, leads to systemic inflammation and organ failure. TLR-targeted therapies aim to mitigate these hyperinflammatory responses. And of course, also toll-like receptors or TLRs have clinical implications. TLR-based therapies are a growing field of research. For example, TLR agonists enhance immune responses as vaccine adjuvants or in anti-tumor immunotherapies. TLR7 and TLR9 agonists are a good example in melanoma. Then we have TLR antagonists, which mitigate hyperinflammatory responses in conditions like sepsis. And then we have, for example, the TLR4 inhibitors, which are explored for treating inflammatory diseases. Also, MHC molecules play a role in diseases and therapies. For example, in immune evasion, viruses like the cytomegalovirus evade detection by downregulating MHC class I expression. But also in transplantation, MHC compatibility is crucial in organ transplantation to prevent rejection, and immunosuppressive drugs often target MHC pathways to improve graft survival. Also, Fc receptors are important for the clinics, for example in monoclonal antibody therapies. For example, trastuzumab targets HER2-positive breast cancer cells and relies on Fc receptor-mediated antibody-dependent cellular cytotoxicity or ADCC for its efficacy. Of course, also T-cell and B-cell receptors are important for therapies. The aforementioned CAR T-cell therapy harnesses modified TCRs to target specific tumor antigens such as CD19, revolutionizing treatment for B-cell malignancies like leukemia and lymphoma. For BCRs, inhibitors of signaling pathways, for example BTK inhibitors, are effective in managing B-cell lymphomas and autoimmune diseases like rheumatoid arthritis. Even not-like receptors play an important role in that context. NLL-targeted therapies aim to regulate inflammasome activity, offering potential treatments for autoinflammatory diseases like gout, type 2 diabetes, and Crohn's disease. Specific inhibitors for the NLRP3 inflammasome are currently in clinical trials to address these conditions. To conclude, immune cell markers and receptors form the molecular language of the immune system, enabling precise communication and coordinated responses. They serve as critical tools for classifying cells, studying immune function, and developing targeted therapies. From CD markers that define immune cell subsets to TLRs that sense infections, these molecules highlight the complexity and adaptability of the immune system. In our next chapter, we will dive into the complement system, a powerful network of proteins that enhances immune defense through pathogen clearance and inflammation. Stay tuned for more insights into the incredible world of immunology. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time, and thank you for listening.